All right, hello, and welcome to your Wednesday lecture on psychoneuroimmunology. That's a mouthful. We've got our sneezing zebra cartoon here today, as we are going to spend some time talking about the interaction between stress and the immune system. All right, so um, we'll start off like we do most times with a clip from Dr. Sapolsky.
20 years ago, Robert got a shocking preview of this idea. The first troop he ever studied, the baboons he felt closest to and had written books about, suffered a calamity. It would have a profound effect on his research.
The Kikarok troop is the one I started with 30 years ago. And they were your basic old baboon troop at the time, and which means males were aggressive and society was highly stratified and females took a lot of grief in your basic off-the-rack baboon troop. And then by now, almost 20 years ago, something horrific and scientifically very interesting happened to that troop.
The Kikarok troop took to foraging for food in the garbage dump of a popular tourist lodge. It was a fatal move. The trash included meat tainted with tuberculosis. The result was that nearly half the males in the troop died.
Not unreasonably, I got depressed as hell and pretty damn angry about what happened. You know, you're 30 years old, you can afford to expend a lot of emotion on a baboon troop, and there was a lot of emotion there.
For Robert, a decade of research appeared to have been lost. But then he made a curious observation about who had died and who had survived.
It wasn't random who died. In that troop, if you were aggressive, and if you were not particularly socially connected, socially affiliative, you didn't spend your time grooming and hanging out, if you were that kind of male, you died.
Every alpha male was gone. The Kikarok troop had been transformed.
And what you were left with was twice as many females as males, and the males who were remaining were, just to use scientific jargon, they were good guys. They were not aggressive jerks. They were nice to the females. They were very socially affiliative. It completely transformed the atmosphere of the troop.
When male baboons reach adolescence, they typically leave their home troop and roam, eventually finding a new troop.
And when new adolescent males would join the troop, they'd come in just as jerky as any adolescent males elsewhere on this planet, and it would take them about 6 months to learn, we're not like that in this troop. We don't do stuff like that. We're not that aggressive. We spend more time grooming each other. Males are calmer with each other. You do not dump on a female if you're in a bad mood. And it takes these new guys about 6 months, and they assimilate this style. And you have baboon culture, and this particular troop has a culture of very low levels of aggression and high levels of social affiliation, and they're doing that 20 years later.
And so the tragedy had provided Robert with a fundamental lesson, not just about cells, but how the absence of stress could impact society.
Do these guys have the same problems with high blood pressure? Nope. Do these guys have the same problems with brain chemistry related to anxiety, stress hormone levels? Not at all. It's not just your rank, it's what your rank means in your society.
And the same is true for humans, with only a slight variation.
We belong to multiple hierarchies, and you may have the worst job in your corporation and no autonomy and control and predictability, but you're the captain of the company softball team that year, and you better bet you are going to have all sorts of psychological means to decide it's just a job, 9 to 5, that's not what the world is about. What the world's about is softball, I'm the head of my team, people look up to me, and you come out of that deciding you are on top of the hierarchy that matters to you.
All right, there's some good words of wisdom there. We have to pick and choose where we find stress in our life. All right, so what is psychoneuroimmunology? It is the relationship between your brain and your behavior and your immune system. Right? And it's interesting because when we think about the immune system is, primarily a peripheral system, right? Set to sort of protect us in the periphery from microbes or toxins, right? But the brain plays a pretty big role in helping regulate that. Right? So psychological states and cognitive processes, things that are happening, stress, can impact the body's defense system. So here you see the cognition here, and then we have microbes and toxins, and we can have impaired nutrition also affect the immune system. Remember, the immune system and the stress system kind of compete for those energy needs, right? So attitude can go a long way. The old saying, laughter is the best medicine, may not be the best medicine, but it certainly is a medicine, right? It is something, having a stress-free lifestyle, having a positive outlook, even in the face of adversity, disease, other challenges, can help you have more positive outcomes, health consequences. So let's dive into the immune system. We have pathogens that are attacking our body, like viruses. We've learned a lot about that this last year. Bacteria, fungi, parasites, less common, right? And we have two general immune system responses. We have a non-specific response, which is inflammation. And so the presence of one of these, of a virus, will cause inflammation. And that allows for increased blood flow, and there are chemical signals that attract phagocytes to the area. Phagocytes are cells that are going to look for things that don't belong, and if they find something they're like, hey, I don't recognize you, I don't think you really belong here, they're going to engulf it and break it down into bits and pieces, and then actually express some of those bits and pieces on MHC2 antigen presenting cells. And that then leads to the second response, which is a specific or acquired response. So we have both chemically mediated and cell mediated responses. So this is where, once a pathogen has been broken down into its little parts, and some of those parts have been expressed as antigen presenting cells, we acquire the ability to recognize those parts of that pathogen, and build antibodies that are going to bind to those parts and signal, hey, this is that thing that doesn't belong here, come get it before it gets out of hand. And then finally, we might need a third stage where we have to boost our defenses with periodic exposures. So a single exposure may not be effective for a long period of time. All right, here is another sort of summary of the immune system. If we look over here on the left-hand side, we see the thymus. The thymus is going to produce T cells, and we'll talk about some different types of T cells. The bone marrow is going to produce B cells. The bone marrow actually produces T cells that are undifferentiated that go to the thymus and become differentiated. So the bone marrow is very important and crucial for our immune system, right? Then we have the lymph nodes and the lymph system. And the lymph system is a circulatory system in our body that is outside our blood circulatory system. So the point of our sort of blood circulatory system is to get oxygen and nutrients to our body. The lymph system is sort of a storage and filtration system of fluid, non-blood fluid. And so B cells and T cells will be densely packed or stored in the lymph nodes. That's why sometimes if you're sick, you have swelling of the lymph nodes. Right? And then release those B and T cells when certain signals that the immune system needs to be active are received. Okay, so here's a figure from your book talking about two different pathways, immune system responses, cell-mediated and chemical-mediated. First, let's talk about the cell-mediated. So here's our macrophage. It's come in contact with an infectious agent. And as the macrophage is devouring, enveloping, getting rid of, killing the infectious agent, it's releasing chemicals that are interleukin-1 and prostaglandin. Now, prostaglandin is going to activate the hypothalamus to turn up our body temperature and reset the body temperature set point from 98.6 up to 100, 101, 102, which creates an unfavorable environment for those infectious agents. It's also going to release interleukin-1, which is going to go activate the T cells. This is the cell-mediated part of it. The helper T cells are then going to release signals to other T cells to activate additional T cells from the lymph nodes, interleukin-2. And those T cells are then going to go and attack the infectious agent. And there's specific killer T cells called cytotoxic T cells that are going to help the macrophages kill these agents. So it's not just up to the phagocytes to get rid of the infectious agents. We can activate this army, if you will, of cytotoxic T cells. Now, as the macrophage is interacting with the infectious agent and activating T cells, there are other T cells that release B cell growth factor, and this is the chemical mediated response. The B cells are going to produce antibodies, and those antibodies then are going to bind to the infectious agent and sort of be like putting a spotlight on the infectious agent, saying, hey, macrophage, cytotoxic T cells, come over here and kill this thing. The macrophage and the killer T cells don't have to recognize the infectious agent. They just look for the antibody, and the antibody is the signal to them that, yes, this is the thing that we're trying to get rid of before it can populate and spread. So, focusing on the chemical mediated response, we have 5 different classes of antibodies. And here we see 4 of those classes, and you can see what each of those classes kind of does. It does a lot of different things. Immunoglobulin D is not listed because we don't really know what the description is. They could have put, IgD and unknown under description, but they didn't do that. So as I mentioned, the B cells produce antibodies. And these antibodies then are like that spotlight that's going to attract the white blood cells to come attack that thing. So this seems like a good time to maybe talk a little bit about vaccines. Vaccines are going to be very important over the next year. And so you should know that there are different types of vaccines. Right? So we can have a live attenuated vaccine, which is basically where you take the actual virus or bacteria and you weaken it so that it can't really spread or populate in the body, and you actually inject that live virus into the body to trigger an immune response and trigger antibody production. So essentially vaccines are training B cells, right? We're putting some part of a predicted virus or bacteria that we may come in contact with in our body so that B cells become entrained, produce memory B cells, and produce circulating antibodies in the plasma. Inactivated vaccines are a dead virus or bacteria. These sometimes need booster shots in order to maintain their immunity. So for instance, tetanus, you're going to need to have a tetanus shot every 10 years in order to maintain that immunity. The subunit or conjugate vaccines is where instead of injecting the entire dead virus or bacteria, we're isolating a piece of the pathogen uh, and injecting that. And this is the way that the Johnson and Johnson COVID-19 vaccine is going to work. Um, some of the other ones that are coming out this year are going to be the subunit conjugative vaccine. And so they're taking like a piece of the COVID virus, just a certain part of it, and injecting that, and then the B cells make antibodies to that part of the virus. And so if you come in contact with the COVID virus, those antibodies are immediately going to latch on to that section of the virus and signal your immune system to attack it right away. So your immune system is sort of primed and ready to go. So vaccines don't actually make you immune to the disease. It's just when you come in contact with the disease, you're able to kill the virus really quickly before it can propagate, before it can infect cells and grow to a level that you become sick. So you actually are infected with the virus if you come in contact with it, but your immune system fights it off so rapidly that you never show symptoms of that, which is why once you do get a vaccine, until we can be sure that the levels of COVID have been knocked down in the population, it's going to be important to still wear a mask, because you could be a carrier. That's what a carrier would be, where you have come in contact with COVID, your immune system is keeping you asymptomatic and knocking it down, but you could still spread it to somebody else. Right? Most vaccines are the subunit type. So you see Hep B, influenza, HPV, meningitis, the pneumonia vaccine, the shingles vaccine. All of those are in that class. That's sort of the most common, biggest class of vaccines. Now, sometimes we want to create a vaccine to something, and the thing we want to create antibodies to, B cells wouldn't recognize it as a foreign entity. So they wouldn't form antibodies. So they have to be sort of stimulated to form antibodies to that thing. So this is an adjuvant vaccine. We're adding an immune system trigger. We're going to stimulate the immune system to recognize this thing it wouldn't normally recognize and create antibodies to it. Well, this is how they are attempting to make a vaccine for Alzheimer's disease. So in Alzheimer's disease, you have amyloid beta 42, which is an amyloid beta amino acid that's 42 amino acids long. Normally, amyloid beta is only 40 amino acids long. And so this amino acid incorrectly is made with 2 additional amino acids, and that makes it useless. It forms plaques in the brain. We think that it's neurotoxic, kills neurons. And so one idea is, hey, can we create antibodies to amyloid beta 42 so that anytime it gets inappropriately made, the immune system would just clear it out, essentially, get rid of it. We would sort of hijack the immune system to be an amyloid beta 42 cleaning mechanism, so that we wouldn't form those plaques, it wouldn't become cytotoxic, and this could potentially reduce prevalence of Alzheimer's disease. Now, the issue is the adjuvant that they used to stimulate the immune system did such a good job at doing that it caused inflammation in the brain and swelling in the brain. The immune system got so activated, and some people died from that swelling of the brain. And so they've had to retool it. So they stopped the trials for about 10 years and tried to find a milder immune system activator that wouldn't have that obviously serious side effect. Finally, we have DNA vaccines. This has been experimental until the Moderna and Pfizer COVID vaccines. The Moderna and Pfizer COVID vaccines are the first DNA vaccines to be released to the public. A DNA vaccine is where they're injecting a strand of mRNA that is going to be taken up by the immune system and cause specific B cells to be made. So there's no like foreign thing that's being put in and 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 B cells are doing their sort of natural thing of recognizing it, differentiating, releasing antibodies. No, we're actually just going in and sort of programming the immune system to create this army of B cells specifically to make these antibodies, which is pretty cool. That's pretty cool stuff. Um, on a side note, I am participating in the Moderna vaccine trial. I received my shots last August and September. And as you know, that vaccine was approved and they showed a 94% effective rate. And so just last Thursday, they brought me in and unblinded the study and told me that I was in the placebo group and then offered the vaccine. So I was able to get my first dose of the COVID vaccine last Thursday. They say it takes 10 to 14 days for that dose to work, and you have about a 70% um, immunity, 70% chance you won't show symptoms. And that's why you need the second dose, which is 28 days later. So on February 25th, I'll go back, get my second dose, and 14 days after that dose, you're at the 94% level. So often these vaccines that you see listed here are going to require more than one dose. Talking about effective rates, everybody was like, oh, how effective is the COVID vaccine? How effective is the COVID vaccine? Well, nobody really asked that about other vaccines. And in fact, you just think, oh, if I get a vaccine, I'm immune and I won't catch this. Well, the pneumonia and HPV vaccines are about 70% effective. That's it. The flu, the seasonal flu, it depends on if they guess right as to what strains of flu are going to be most prevalent. But in general, those range from 40 to 60% effectiveness. Better than nothing, but certainly not 94%. And there's a difference in the way the DNA vaccine can activate and generate these B cells. It really ups the level way higher than the conjugative vaccines do. So, for instance, the Johnson & Johnson COVID vaccine, that's the conjugate, is only 66% effective against COVID, whereas both Moderna and Pfizer are in the mid-90s. So there's your lesson on vaccines. I think it'll be very important to understand this as we move forward. If you have a choice of a vaccine, which one would you prefer? Probably an mRNA vaccine than the Johnson & Johnson. Now, if the Johnson & Johnson is what's available and that's what you can get, certainly take it. 66% is better than 0%. All right, so let's look at the cell-mediated response. That was the chemical-mediated response. Cell-mediated response, we have these T lymphocytes that, as I mentioned, originate in the bone marrow, and then they are going to mature or differentiate in the thymus. They primarily defend against viruses, so here we can see sort of what happens on in that sequence on the left-hand side, right? There are different types of T cells. And so we have memory T cells. These are formed after the infection. And so they remember that virus and how to recognize and kill that virus. There are helper T cells. These are releasing those cytokine interleukin chemicals that are activating B cells and other white blood cells. And we also have some helper cells like the APC, the antigen presenting cells that are going to help transfer antigens to helper T cells. The lymph node system is involved in releasing those dendritic, APC dendritic cells. And then finally, the killer T cells, the cytotoxic T cells, are the ones that are actually going to kill the pathogens. And they do this by sort of drilling holes in the pathogen, if you will, with a substance called perforin. They perforate the cells. All right, so I mentioned the antigen presenting cells. We need to talk a little bit about antigens, right? Major histocompatibility complex, MHC. MHC1 molecules are expressed on the surface of every cell in our body. And it's sort of like, hey, I'm this cell and this is what I do. I'm a red blood cell and I carry oxygen and I'm part of the circulation system. And so our immune system reads this code, if you will, and it knows, okay, that MHC pattern is my pattern. That's one of my cells. Don't mess with that. Or I don't recognize that MH1C cell. That's a foreign thing. That's a virus. And so I'm going to kill it, right? MHC2 molecules are what is made by these professional APC cells. And they are markers, if you will, of the virus, right? So when the macrophage envelops the virus and breaks it down into smaller parts, it'll take some of those proteins or parts of the virus's cell surface, express them as an MHC2 molecule on the surface of the macrophage, and then dendritic cells will help get that to B cells and T cells to know, hey, this is what you're looking for, right? Make antibodies for this thing. So we know that the code for MHC1 molecules, the things that are naturally expressed on our cells, the genes are located on chromosome 6. And there's an unusually high number of alleles. So the patterns then become very individualized. No two people have exactly the same patterns. Now, the closer related you are to somebody, right, the closer your alleles will be, and the less related you are to someone, the chances are that you're not as compatible. So this is the problem with tissue transplantation and tissue typing, is that what we're trying to do if someone needs a bone marrow transplant, right, they are looking for someone who has the closest pattern, MHC1 molecule pattern, to that person to be the donor, because the closer you are to that pattern, the less likely the tissue will get rejected, right? All right, so that's a little bit about transplantation and why typing of the tissue matters. It's all because of this MHC1 pattern that's pretty individualistic. Okay, so that's sort of the immune system in general. Now let's talk about things that can manipulate the immune system. And serendipity in science is kind of like PB&J. They just go together, right? And so often in science, we sort of serendipitously discover something by accident. And that was the case for this next experiment. Some researchers were looking at conditioned taste aversions, and they were using a chemical called cyclophosphamide as the aversive thing that was going to cause a taste aversion to saccharin, right, which is a sweet taste. And it turns out that this aversive drug, cyclophosphamide, is an immunosuppressant. It suppresses the immune system. So rats were given pairings of these two things, repeated pairings, and then they were tested they gave them saccharin to see if they learned to avoid the saccharin because it was paired with the immunosuppressant. And what was interesting was not necessarily that rats learned a taste aversion, but as rats continued to receive saccharin in absence of the drug, in absence of the immunosuppressant, their immune system continued to be suppressed. So in addition to the taste system learning to associate being the negative effects of the drug with the sweet taste, the immune system learned to associate being suppressed with the saccharin. And so this was interesting because it gave a rationale for why we might have allergies, right? So why should I be allergic to peanuts? I mean, I'm not, but someone who is allergic to peanuts, why would they be allergic to peanuts? Peanuts aren't bad for you. Peanuts shouldn't trigger an allergic response. Well, if an immune response somehow got associated with that taste with peanuts, right? Then the peanuts might trigger the immune system, which would cause the allergic response. So it gave us sort of an explanation for how someone might come to learn an allergy. Okay, input to the central nervous system. So how does the immune system and the nervous system communicate with one another? And so there's a couple of ways. And some of this, I'll just warn you, is not, we don't fully understand maybe all the different ways in which they can communicate. So one of the jobs of the spleen is to detect bloodborne antigens. The spleen is innervated by neurons, and this is a possible neural pathway back to the central nervous system to say, hey, the spleen detected this. This is circulating in my body right now. Cytokines are probably the primary way that it's sort of a hormonal chemical communication. And they can act both on the vagus nerve, cranial nerve 10, to send input into the brain. And they can also act on the hypothalamus. So interleukin 1 or prostaglandin is a cytokine that causes fever in the hypothalamus. And the vagal nerve can also trigger vomiting. So if we think that we're sick and maybe it was because we ingested something poisonous, we would trigger that vomiting reflex. That's why you may be nauseous or vomit when you have the flu or some other virus, even though you didn't eat anything, and vomiting isn't going to help get rid of the virus. It's sort of all linked together in this system. Okay, output from the central nervous system. And so we think the way the central nervous system communicates or helps control the immune system is through the HPA axis, right? So now we're introducing stress into the system, but here we're actually, being sick is a stressful sort of condition, right? But and that's then going to kind of regulate energy needs. So if we trigger a small stress response in response to the immune system being activated, not a big stress response, but a small, amount of glucocorticoids being released, what is that going to do? Well, it's going to increase blood flow, which is going to move the T cells and B cells and antibodies around the body better. It's going to increase blood glucose so that the immune system has energy needs, right? And so those are both positive things. Now, we'll see that glucocorticoids are also provide a negative feedback, right? So they can suppress the immune system, particularly when they're released for a long period of time, like when you're sick. And so they're sort of keeping the immune system from going off the rails, right? From being too active while you're sick. Right? And then once you're able to eliminate all of that and the immune system's no longer being triggered, it'll sort of help shut it down. Okay. So responses to being sick. We just talked about HPA axis. We talked about fever induction through the preoptic area of the hypothalamus. Talked about vomiting through cranial nerve 10 to the NST or the NTS and area postrema in the brainstem. And then we have sort of decreased motility and decreased appetite while we're fighting this off, right? We're trying to just let the immune system do its thing and send all the energy we can there. And not expend energy in finding food or eating. And again, a lot of this is through the interleukin-1 cytokine signaling pathway. That's what that IL-1 on that diagram is there. All right, so father of stress, Hans Sell. He was the first one to sort of identify the stress response, and in rats. And I don't think I told that story in when we did the overview of the stress system. But the thought. So again, serendipity and science. Hans Sell was working with an endocrinologist on effects of estrogen, and he was injecting estrogen into rats. But as the story goes, he wasn't particularly good at injecting the rats. And so sometimes he'd drop the rats and have to chase them around, or he would have a bad injection. And so essentially he gave all these estrogen injections to the rats, and then saw all sorts of effects that were stress mediated, but he didn't know they were stress mediated. He thought, oh, look, hyperestrogen causes atrophy of the thymus. It causes cardiovascular disease. It causes all the effects that we see, suppression of the immune system. All these effects that we see in stress. And so he did the control where he's injecting rats, again, poorly, with salines, and lo and behold, they also had all these effects. So then he's like, oh, it must be the stress of this experiment that's causing this, right? So the thymus is important in that T cell differentiation. And so stress, glucocorticoids can impair the ability to make those T cells, those important T cells. We also see decreased white blood cell lymphocyte secretion, decreased circulation, length of circulation. So sort of the immune system just doesn't become as robust. There's a decrease in antibody production. The inflammation is suppressed. So sometimes when you're sick, you'll get put on a steroid, and that steroid is to reduce the inflammation. Well, glucocorticoids are a steroid, and so they can suppress that inflammation as well. And so then that doesn't attract the phagocytes as much, and sort of muting the immune system response. And glucocorticoids can also actually trigger an apoptosis pathway in lymphocytes and kill lymphocyte cells, cause lymphocyte cells to die. That also interferes with interleukin and these cytokines and interferon release, and so that makes lymphocytes less responsive because we're sort of damping down everything, right? And as I mentioned, interleukin-1 triggers corticotrophin releasing factor, or hormone, which is then going to trigger ACTH, which is then going to trigger corticoids in this sort of negative feedback to turn down the immune system. Now, when we have someone who's been chronically stressed, right, then that's going to weaken the immune system before it's even activated. All right, so here's Hans Sell's 3 stages of stress that we talked about. But now I want to add in what we didn't talk about is, okay, what happens in the immune system in each of these? So we have our acute stage, our alarm stage, which is sort of the normal response. And so normally, in response to a stressor, there's activation of the immune system, followed by deactivation, suppression of the immune system to a return to normal if that stressor is gone and we just have our adaptive normal stress response. However, when we enter into that chronic or resistance stage, we're now getting chronic immune suppression, and we can suppress the production of antibodies and lymphocytes, and through that lymphocyte apoptosis, show an overall suppression of our immune response by 40 to 70%, which is a lot, right? Really weakening that. So now when you come in contact with a virus, you don't have adequate circulating levels of antibodies. or T cells to fight it off. And then that can lead to exhaustion in the last stage. Okay, so I want to go into a little bit more detail about this immune system activation and deactivation by an adaptive sort of normal stressor. And so when we say the onset of stress increases immune function, that's primarily done by the initial release of norepinephrine and the catecholamines, stimulating that increase in the immune system. And the turning off is primarily done by the glucocorticoids, right? And so that phase B is really critical, the presence of the glucocorticoids to sort of shut down the immune system response. So one way that we could induce autoimmune disease is to have a failure of that phase B. And that would then just be activating the immune system, and it could get high enough that it starts to recognize the MHC1 molecules of our own cells and attacking them, recognizing them as foreign antigens, not antigens that belong there. And so having inappropriate glucocorticoid response, either blocking that glucocorticoid response in rats, can increase the autoimmune disease risk. In fact, Sapolsky talks about in the book cases of sort of spontaneous autoimmune disease in animals. It turns out that they generally have something wrong with their glucocorticoid response, an inability to shut down the immune system. So far we have two effects of stress in the immune system. Chronic stress can suppress the immune system and not allow it to be effective, or not having the appropriate glucocorticoid response to an acute stressor could allow autoimmune disease to occur. But what about that really common stressor, chronic intermittent stress? What if we just activate phase A, phase B, go back to normal, and keep activating phase A, phase B, phase A, phase B, right? And at the same time, we're decreasing the effectiveness of the brakes, we're decreasing the parasympathetic nervous system. And what could happen with these chronic intermittent little stressors is that we don't return back down to baseline, right? The B phase gets a little weaker each time. And so slowly with chronic intermittent stressors, we are slowly creeping up towards that autoimmune disease range. Okay, so here's an example of how an autoimmune disorder could function, and this is called Graves' disease. And in this particular autoimmune disorder, it's not that the immune cells are attacking something, it's that there's too much IgG antibody, immunoglobulin G antibody, being produced. It's overproducing that, and this antibody activates the thyrotropin receptor, which is a thyroid stimulating hormone receptor in the thyroid. And so it's artificially activating the thyroid and causing a lot of T3 and T4 to come and circulate throughout the body, right? Because of a negative feedback loop, right? It's also going to cause natural thyroid stimulating hormone to decrease, but that doesn't really matter because the antibody is triggering the release. So we have the THS levels falling because of the negative feedback loop, but the thyroid's just raging and producing these T3, T4. So we get very high levels of circulating thyroid hormones, and we're damaging the brakes of this other system, right? So this can result in an enlargement of the thyroid gland that can become visible as a goiter. So this is located in your neck. You see the doctor down there examining the woman that has an enlarged thyroid. Thyroid hormones are used to regulate our metabolism and our growth. They also can have an effect on the catecholamines. So symptoms include increased heartbeat, muscle weakness, disrupted sleep, irritability, all sorts of issues like that. Again, example of an autoimmune disorder. Another example of autoimmune disorder is multiple sclerosis that we talked about. That disease is where the immune system is attacking cells, myelin cells in particular. And so there are different ways that autoimmune disorders can happen, but essentially the hallmark of all of them is that the immune system is hyperactive in an inappropriate way. All right, so there are two routes, if you will, that can lead to compromised immune systems or altered survival rates. The neuroimmune route we've been talking about, right? Individuals experience stressors, chronic or intermittent, inappropriate release of glucocorticoids. These glucocorticoids then suppress the immune system, change our disease resistance, make us more likely to become sick or fight off disease. There's a second route that could happen through lifestyle changes or lifestyle impacts that may not involve stress per se, right? So if you are isolated, if you are lonely, if you are bereaving, all of these things seem to have a negative impact on our immune system and may involve stress, but may not involve stress, right? And so it can cause changes in behavior like not taking preventative measures and increasing your risk factors. I'm going to take this minute to say that I'm not going to cover the section on cancer that Sapolsky has in this, but I would like you to read that and cover that on your own. It's pretty self-explanatory, but basically talking about some of this altered risk or increased susceptibility may change the prevalence of cancer or cancer survival. All right, so not surprisingly, if you take rats and you shock them, you get a suppression of the immune system from the stress of being shocked, right? We get decreased circulating lymphocytes, increasing stress, decreasing lymphocytes. If we adrenalectomize an animal, meaning they can't produce glucocorticoids, we prevent that decrease from circulating lymphocytes, right? So there's no glucocorticoids to kill the lymphocytes levels stay high. Now, but here's the interesting thing. If we look at the stimulation of those lymphocytes, like how well can those lymphocytes attack an antigen, it turns out in both groups, even the adrenalectomy group, shock compromises that, right? So, and so there are non-stress related events. So when an animal is put in a negative situation, like being shocked, there are non-glucocorticoid effects that happen that weaken the immune system's response. I talked about bereavement, right? So long-term stress can affect our immune system resources. So again, looking at the ability of, if we look at, we're increasing the amount of antigen that we're adding to a culture, and we're seeing the immune response go up. And we did this before a husband's wife died, and then looking at the husband's lymphocyte responses afterwards, right? And we see this decrease, right? And that could happen through specific mechanisms like decreasing the functionality of the thymus, the spleen, and bone marrow. In fact, bereavement can be so strong that if you're in a compromised position, so we've talked a lot this last year about people who are at risk individuals. And who are those people? Who is currently getting the COVID vaccines, approved to get the COVID vaccines? It's people at the highest risk. And who are they? People, healthcare workers who are going to be exposed a lot to the virus, and elderly. Because when we're elderly, our immune system is already compromised. Our ability, if we do get sick, it's a lot harder to fight it off. Our body has a harder time dealing with that. So bereavement, when do people usually lose their spouses, especially if they've been married for most of their life right, and they're elderly and you lose a spouse, it's a major life-changing event. It can be, and it can be catastrophic. And in fact, broken heart syndrome is a real thing, right? It's not uncommon at all that when one elderly spouse passes away, that they're followed not too long behind by the other spouse. And so here's a clip just from a few years ago where George Bush passed away several months after his wife had died. The New England Journal of Medicine has looked at this, and there are effects on the heart tissue, like you actually can break your heart due to sudden emotional stress, right? The myocardial stunning due to sudden emotional stress. There's impaired circulation capacity in absence of other cardiac damage, right? No evidence of that. There's also 7 times the amount of circulating catecholamines in people that are bereaving, which means a lot of norepinephrine and epinephrine and dopamine being released. This typically has a short duration, but it's going to depend on the bereavement intensity and period, and the longer, the harder it is on the body. This hasn't fully been, vetted out and figured out, but it definitely looks like a real thing, something that we've just recently sort of discovered, right? People have probably known about it for a long time. All right, that's what I'm going to cover for today. Good lecture. I'm looking forward to talking to you on Friday for our research discussion, and I will see you then at 10:30 on the Zoom.